going to see about the level of organization of a gene. There are about 5 levels of organization to be occurs. Now we are going to see this 5 level of organization makes it to be chromosome. From the DNA, it is converted into chromosome by this 5 level of organization. Now we are going to see about each level of organization individually. First, the first level of organization. It is going to be known as nucleosome organization or bead and string model of organization. It is known to be nucleosome organization, bead and string organization because here the bead is going to be a octamer histone. Octamer histone means it consists of H2A, H2B dimer on top and bottom, H3 and H4 tetramer at the central. This is going to be a bead. String means DNA. This DNA is going to wound around the histone octamer. Therefore, it is found that like a bead, there is an octamer to be present. Like a string, the DNA is going to be wound around it. This type of organization is going to be taken place. This method of organization is known to be a nucleosome. So, a DNA is going to be associated with the histone proteins. This is known as nucleosome. Here, a DNA is going to be wound around it. This wounding is found to be 1.65 times around the core protein. That means the DNA is going to be wound around the protein about 1.65 times. This nucleosome can also be disintegrated. This is not a stable form of organization. This nucleosome can also be disintegrated by micrococcal nuclease. A bacterial enzyme like micrococcal nuclease can digest this nucleosome. Next, we are going to see about the elements of bead and string model or nucleosome model. As we said that a DNA is going to be acts like a string, an octamer of histone is going to act like a bead. This is going to octamer of histone itself known to be core histone and linker histone, which is not involved in this. A core histone only be going to be speak in this model, where a DNA can also be known as core DNA and linker DNA. So, a core DNA is a DNA which is going to be wound around the octamer. This DNA is going to be a core DNA. A linker DNA is going to be a DNA which is away from the octamer. This is going to be a linker DNA. So, the core DNA consists of 146 base pairs of DNA. Therefore, the 146 base pairs are found in the core DNA, which can be digested by the nucleus. A linker DNA is going to be 8 to 114 base pairs. In per nucleosome, the linker DNA is consists of 8 to 114 base pairs. A core protein, as we said already, it is an octamer protein which is going to be 6 nm height and 11 nm in diameter, which means so this octamer is going to be found which is going to have a 6 nm height, which is 6 nanometer in height, and the diameter is going to be 11 nm. The DNA is going to be wound around it, which is going to have 146 base base pairs in it, and the linker DNA is going to be 8 to 114 base pairs in it. This is the structural form of nucleosome to be present. Here it is a structure. This is the nucleosome structure. So it is a core DNA. This is going core protein. This is going to be a core DNA. Between the two nucleosomes, there is a DNA present, right? This is going to be a linker DNA. This linker DNA is going to have the 8 to 114 base pairs, and the core DNA is going to have a 146 base pairs to wound around it. So it is 8 to 114 base pairs. It varies between these base pairs. Here I have mentioned that linker histone also. The linker histone is going to make the two nucleosomes to attach. It is going to attach with the linker DNA and make the new nucleosome to attach into it in the second level of organization. Here we are going to see only the first level of organization, where there is a nucleosome concept, where there is a core histone, which is a histone octamer. For core DNA it is wound around it, and there is a linker DNA. So as we said that about 1.65 times the turn must be there. So these 1.65 times of turns are going to consist of 80 base pairs of DNA. Among the 114 base pairs to wound around the histone proteins, 80 base pairs of DNA are going to have a turn of about 1.65 times. A degree of supercoiling for the mini chromosome. It was not important, but I have explained here, because a supercoiling may happen when there is no octamer or no histone proteins to be occurs. When there is no histone proteins, the coiled form of the nucleosome form of DNA is going to be relaxed off and can supercoil to minus 1.65 times. This supercoiling of DNA without the protein. Without protein, the DNA may supercoil to minus 1.65 supercoiling. This is known as linking number paradox. This supercoiling of DNA without protein is known as linking number paradox. Therefore, the torsional strain in the protein makes it to prevent it from supercoiling. Therefore, a nucleosomal DNA has a torsional strain, which makes it to which turns can prevent it from supercoiling. Here we can see the turns. A DNA is going to be attached with the octamer protein, which is histone. As we said that histone is going to be 6 nm height and 11 nm in diameter. This is a central region. So the histone, a DNA is going to be wound around it of about 2 nm. From the histones, there are about 2 nm diameter region is covered by the DNA, which is going to be made by torsional strain. Apart from as the basic character, which is positively charged due to the lysine and arginine, and the DNA is negatively charged due to its phosphate group. This charged balance makes it to wound around it. There is also a torsional strain which makes it to wound around it. Without the histone proteins, it may undergo supercoiling and makes it to disintegrate. As the supercoiled mini chromosome can also be relaxed by topoisomerase enzyme. There is a mechanism too, but relax, histone proteins are used and makes the torsion strain to prevent it from supercoiling. But this topoisomerase is going to be used during the replication process when the DNA wounding is going to be relaxed. It may undergo supercoiling. This supercoiling can be prevented out by topoisomerase enzyme during the replication process.